Welcome to our fourth and final in the series on endocrine and cell communication. Today we'll be considering maintaining balance or homeostasis. Remember, in our AP Biology curriculum framework, we're considering the topic of cell communication. Today we'll be looking at that long-distance communication using chemical signals. And we'll continue looking at endocrine cells and endocrine hormone production, particularly looking at insulin and its paired hormone glucagon. We'll also mention calcium regulation and the hormones that are involved with that. Let me call your attention to the learner objective that's included in the AP Biology curriculum framework that says you will be able to use representations or models to analyze both quantitatively and qualitatively the effects of disruption to dynamic homeostasis. So I want you to listen for those disruptions and those effects today as well. As a means of brief review, let's consider what would make a chemical be called a hormone. What are the attributes that a chemical has to earn the label hormone? Well, first of all, it has to be produced by an endocrine cell. And then that chemical would also have to be transported through the body by means of the bloodstream. And then our third characteristic for a hormone was that hormones have target cells. Those target cells are identified by the fact that the cells have receptors for those particular chemicals. So in a simple hormone pathway, hormones are going to travel from the cell that secretes them to target cells who contain their receptors. So you'll need to hang on to that information as we move through today's examples looking at our paired hormones. If we were to start with a really simple hormone pathway, let's consider what happens when you eat. You swallow your food, you chew it, it goes into your stomach, and in your stomach your food's going to be mixed with the acidic conditions or the acidic solution, and the stomach is lined to be protected from acid with a thick layer of mucus. Now that acidy chyme is going to move into the small intestine. The small intestine doesn't have that same adaptation to protect it from acids, and so the acid serves as a stimulus for the endocrine cells that are found in the small intestine. They're called S cells, and they will secrete a hormone called secretin. When secretin is picked up by the bloodstream, it has a target cells that are in the pancreas, and those cells are going to release bicarbonates, and the bicarbonates, as shown in this diagram, the bicarbonates then that are released are the response that actually causes the condition to be more alkaline instead of acidic, and that would stop this stimulus. So here's a typical negative feedback pathway where an endocrine cell secretes a hormone. The hormone, in this case secretin, is circulated by the bloodstream to target cells, in this case pancreatic cells. Those pancreatic cells have some sort of response. In this case, it is they release bicarbonate ions into the small intestine, and that action is going to stop the stimulus because the once low pH now becomes an alkaline pH, and so this pathway is slowed down or even stopped with its production. And so we see that typical hormone pathway oftentimes results in a negative feedback loop that is self-regulating to help maintain homeostasis, which is so important in all of our body systems. So you'll see that negative feedback loops play a critical role in the maintenance of homeostasis, and secretin is just one example of such a pathway. In addition to the negative feedback loops, we also have positive feedback occurring in our body. One clean example of that would be in mammals, the hormone oxytocin causes the release of milk. That is going to cause the baby to have greater suckling, and then that's going to stimulate the production of more oxytocin. So we see an amplification of the pathway rather than a stopping of the pathway. And so those sorts of regulatory mechanisms are called positive feedback loops. You see oxytocin could also be involved in a positive feedback loop during the birthing process. When oxytocin is released from both the fetus and the mother's posterior pituitary. It causes the uterus to contract. The more the uterus contracts, then the more pressure that's going to be exerted, which is going to cause the pituitary glands to release even more oxytocin. And so we see this loop that causes more and more contractions until eventually we have the birthing of the baby. And so that's a classic positive feedback example that we see. Let me take your thoughts back now to negative feedback. We're going to look at an example where two hormones, insulin and glucagon, work together as a tandem pair to maintain blood glucose levels. This pair of hormones work together to keep our levels homeostatic. Insulin's role is basically to decrease the amount of glucose in the bloodstream. It does so by allowing glucose, or affording glucose the opportunity to enter through the cell membrane. Glucagon, on the other hand, has the role of, when glucose is gone, it will serve as a method or mechanism of increasing the glucose level in our bloodstream. It mobilizes fat to be broken down as well as glycogen to be broken down to increase the amount of glucose that would be in our bloodstream. Both work together then to maintain homeostasis. The cells that produce these two hormones are found in the pancreas. There are alpha cells in the pancreas, and they produce glucagon, the hormone. The beta cells of the pancreas will produce insulin. So the pancreas has two different types of hormones that are produced that touch the process of blood glucose level regulation. And the hormones that we would consider then are insulin and glucagon. I'm going to show you a short video clip that explains the relationship of these two hormones to one another, and hopefully will help you understand how this antagonistic pair of hormones can help maintain homeostasis.
After a high carbohydrate lunch, like a plate of spaghetti, polysaccharides are digested into monosaccharides. After a high carbohydrate lunch, like a plate of spaghetti, polysaccharides are digested into monosaccharides.
All right, let's watch this video clip that shows the relationship between insulin and glucose, and then also the hormone glucagon. So let's look at how those work together to maintain a relatively constant amount of glucose in our bloodstream.
After a high carbohydrate lunch, like a plate of spaghetti, polysaccharides are digested into monosaccharides. Monosaccharides, such as glucose, are absorbed by the small intestine and released into the blood. Increased levels of blood glucose signal the pancreas to secrete insulin into the bloodstream. Insulin promotes the uptake of glucose by most cells of the body. Many cells, like muscle, burn glucose for their metabolic fuel. Fat cells in adipose tissue use glucose to make fat. Liver cells convert glucose to glycogen and fat. As the afternoon passes, the cells continue to take up glucose, and blood glucose levels decrease. By 6 o'clock, all the glucose from the spaghetti lunch has been absorbed, and blood glucose levels have fallen further. The low blood glucose puts a brake on insulin release from the pancreas. Without a glucose supply, cells switch to using glycogen and fat as their fuels. The liver breaks down glycogen to glucose and converts fat to fatty acids. Fat cells convert fat to fatty acids. These fatty acids are used by other body cells as fuel in place of glucose. The glucose released from the liver is reserved for nerve cells, which require a constant supply.
So that shows us an explanation of how insulin works in relationship to the cells. Let's look inside a single cell and see how insulin impacts the activities of that cell. When you look at this graphic, you can easily see a simple fluid mosaic membrane that's got a bilayer of phospholipids. You also readily will notice that there are integral proteins spanning the width of this membrane. Here's the inactive version and then the active version of that particular protein. And then there's another protein, another type of integral protein here on the end, on the right side. Let's start with the left side protein. This protein that's integral or embedded in this membrane is serving as an insulin receptor site. The hormone insulin has come from the bloodstream and has arrived on the outside of the cell. When it binds with its receptor, you'll notice that there is an overall change in the shape of the receptor and an activation of a series of proteins that are going to result in the presence of a transport protein that will facilitate the diffusion of glucose from outside the cell to the inside of the cell. So the glucose was brought to the cell by the bloodstream. It could not enter on its own because it's too large. This membrane is impermeable to glucose, but the diffusion of glucose from one side of the membrane to the other is facilitated by this protein that has been activated from the presence of insulin. So there's a chain of events that occurs when insulin arrives at a cell and docks with its receptor, ultimately resulting in the movement by diffusion of glucose from one side of the cell membrane to the other. So now this receiving cell has enough glucose to provide the energy needed for cellular activities. So if we consider this graphic, remember the learner objective said that you would be able to describe representations of how homeostasis is maintained. You might want to stop the video for a moment and explain this orally to yourself or to a partner, because this graphic also includes what happens when the blood glucose level declines, as well as what happens when the blood glucose level is high. And so you'll see two hormones identified here, both insulin and glucagon, which work in a tandem pair to be antagonistic toward each other. So their effects oppose each other. And together they work to maintain our blood glucose level at somewhere between 70 to 110 milligrams per 100 milliliters. So stop the video for a moment and describe the relationship between insulin and glucagon. I bet your description sounded something like this. If your body detects an increase in blood glucose levels, the beta cells in your pancreas are going to release the hormone insulin. It's going to be transported by the blood system to individual body cells who need that glucose for normal cellular activity. So they will be able to uptake glucose if insulin is present. Some of the glucose is going to be taken in by the liver when insulin is present. The liver will take up the glucose and store it in the storage form of glycogen. That way it can be readily accessed should there be a need for additional glucose at a later point. Both of these work together to cause a decline in the blood glucose levels, which would then return our system to homeostasis, putting our blood glucose levels back to an acceptable level. But if you were to skip a meal and your blood glucose level drop, if we skipped a meal, then what's going to happen is the pancreatic cells that are called alpha cells, those alpha cells are going to release glucagon, which is the antagonistic hormone to insulin. And when glucagon is released, glucagon is sort of the opposite of insulin. When glucose is gone, glucagon is going to stimulate the liver and adipose cells, don't forget the fat cells, those adipose cells will release glucose, and when they do, that's going to cause glucose levels to rise to a more homeostatic condition. So we release glucagon when we are low in blood level glucose, and we release insulin when we are high, when we have high levels of blood glucose. And so together, these two help regulate the swings in the drops and the rises of our blood glucose level to keep us at that homeostatic condition. The process you just described happens when things are working normally or as expected, and there's an appropriate signal transduction pathway in place. But sometimes things don't work as designed, as is the case with diabetes. Now that we've seen the role of insulin, you can think about why a lack of insulin could lead to elevated levels of glucose in the blood. Think about that with your partner for a minute. Talk about it. Can you explain why a lack of insulin would result in elevated blood glucose levels? And why might that be harmful? High blood sugar can make you feel tired. It can damage your kidneys. It can even cause blindness. So diabetes is a fairly serious disorder. Even though it's common, it's very serious. It has two basic forms. In type 1 diabetes, it's also known as insulin-dependent diabetes. It's an autoimmune disorder, or it is the result of an autoimmune disorder. And that disorder destroys pancreatic beta cells. And remember, those are the cells that go through the protein synthesis and produce insulin, the hormone. And so if they are damaged, they can't produce enough insulin to maintain the need that the individual cells of the body have. In type 2 diabetes, or non-insulin dependent diabetes, this involves an insulin deficiency, or maybe the cells can't respond properly. The response portion of the signal transduction pathway is interrupted. There could be a change in the receptors and they're not working properly. So when that system is out of balance, you get these conditions like diabetes. It's one of the more prevalent endocrine disorders in the United States and in Northern Europe. Some 17 per 100,000 individuals will express diabetes. In Scandinavia, it's even higher, with 35 people per 100,000 showing the disease. Where, by contrast, Japan and China have a much lower count, with only one person per 100,000 that exhibit this disorder. Let's consider one more chemical, calcium, whose level needs to be maintained at homeostatic conditions in the human bloodstream. The gland involved this time will be the parathyroid gland. Located just behind the thyroid gland, the parathyroid gland produces the parathyroid hormone. Don't you love it when they do that in biology? They named the hormone after the gland. That's nice. The parathyroid hormone's job is to stimulate the calcium uptake by kidneys, so you start to save more calcium from your bloodstream, as well as to stimulate the release of calcium from your bones. That's our primary reservoir of calcium in our body. And so parathyroid hormone helps release calcium from our bones. Parathyroid hormone helps take up calcium by the kidneys. It also activates vitamin D, sets off a chain of events which is ultimately going to result in the absorption of additional calcium by the intestinal tract. So through those three venues, we're going to have an increase in the calcium that's put back into the bloodstream. So naturally, our blood calcium levels will rise back to that homeostatic condition. So the blood calcium levels can be maintained at a homeostatic condition by a pair of hormones, the parathyroid hormone and calcitonin. Remember, parathyroid, as I said, works to also activate vitamin D that plays a role in this calcium absorption into the bloodstream. So parathyroid is going to get our intestinal tract to save calcium, our kidneys to save calcium, and then also take calcium from our bones when our bloodstream levels are too low. Now, if our bloodstream levels are too high and too much calcium is found in the circulatory system, then the hormone calcitonin is secreted by the thyroid gland, and it will decrease the levels of calcium by putting calcium into the bones and then allowing the secretion of calcium out of the body by the kidneys. So these two work together in order to maintain a constant condition of calcium in our bloodstream. So let's look at a description of how calcium levels are regulated in our body.
Calcium levels in the blood are regulated by three hormones: calcitonin, parathyroid hormone, PTH, and vitamin D. When calcium levels rise, the hormone calcitonin acts to reduce blood calcium. When calcium levels are too low, PTH and vitamin D act synergistically to increase blood calcium. The thyroid gland, which sits atop the trachea, produces the hormone calcitonin, while the parathyroid glands, located on the back of the thyroid, produce parathyroid hormone. Calcitonin and PTH maintain calcium homeostasis by controlling the deposition and absorption of bone, the excretion of calcium by the kidneys, and the absorption of calcium by the digestive tract. Elevation of blood calcium levels triggers certain cells in the thyroid gland to release calcitonin into the bloodstream. Calcitonin acts to lower the concentration of calcium in the blood through its effect on bone, where 99% of the body's calcium can be found. Two cells in the bone act to regulate bone production and destruction. Osteoblasts take up circulating calcium and deposit new bone, whereas osteoclasts break down the bone and release calcium into the bloodstream. Calcitonin decreases the activity of osteoclasts and thereby shifts the balance of bone turnover to favor deposition of new bone and removal of calcium from the blood. Calcitonin's ability to regulate bone formation and resorption makes it an important regulator of calcium levels in many animals. However, calcitonin does not play a major role in calcium homeostasis in adult humans. If the thyroid gland is removed from an adult, the body is still able to regulate calcium levels in the blood. In contrast to calcitonin, the parathyroid hormone exerts a broad influence on blood calcium levels. Calcium levels are sensed by receptors in the plasma membranes of the parathyroid cells. When these receptors are activated, they inhibit the synthesis and release of PTH. A fall in the blood calcium levels removes this inhibition and triggers the synthesis and release of PTH. Parathyroid hormone acts in a number of ways to increase blood calcium. Like calcitonin, PTH targets bone cells. PTH binds first to osteoblasts, causing them to release cytokines. The cytokines increase both the number and activity of osteoclasts, thereby enhancing bone turnover. Overall, there is a net loss of bone and a rise in blood calcium levels. PTH also conserves calcium by stimulating the kidneys to resorb it rather than losing it in the urine. Finally, increased secretion of PTH causes the digestive tract to absorb more calcium from food, but this is an indirect effect dependent on vitamin D. In the kidney, PTH stimulates the conversion of vitamin D to an active form, and it is vitamin D that acts on the digestive tract to enhance absorption of dietary calcium. The lipid-soluble vitamin D enters epithelial cells in the intestines and combines with a cytoplasmic receptor, forming a transcription factor. In the digestive tract, this transcription factor acts to increase the synthesis of calcium pumps, calcium channels, and calcium-binding proteins, all of which promote the uptake of calcium by the intestinal epithelia and its transport into the bloodstream. In the kidneys, vitamin D acts with PTH to reduce calcium loss in the urine. The actions of vitamin D in bone are more complex. In the short term, vitamin D directly promotes bone turnover, liberating calcium into the blood. In the long term, however, elevated calcium levels promote the deposition of new bone and the removal of calcium from the blood. Vitamin D also acts on parathyroid cells to inhibit the transcription of the PTH gene, thus forming a negative feedback loop for the regulation of PTH. Overall, the concentration of calcium in the blood is tightly regulated by the combined action of calcitonin, PTH, and vitamin D.
But if you were to look at the relationship between the parathyroid hormone and calcitonin, with vitamin D aside, looking at the role these two play in the maintenance of homeostasis, could you take these labels here on the right and indicate which portion of this representation each statement matches to best? Take a moment to do that. You may want to stop the video so that you can freeze this image, or your teacher may have provided you with this image. Either way, take a moment to practice looking at the relationship between blood calcium levels and these two target hormones. Your results hopefully look something like this, where if we start here at 1, blood calcium levels rise, calcitonin, your solutions may look something like this, where if we start with a rise in blood calcium level, the thyroid gland produces calcitonin, which stimulates the deposition of calcium into the bones, causing a lowering of blood calcium levels. If calcium levels drop too low, then they fall below that set point, and our parathyroid gland would produce parathyroid and release parathyroid hormone, which is going to cause our blood calcium levels to rise above the set point, and that process can return to homeostasis, or we may see again an increase in the calcium levels followed by a decrease when these two hormones are working properly. Hopefully your solutions look something like this. We're at area number 1 in this representation. It shows or indicates a blood calcium level rise. When that happens, the thyroid gland here, shown at number 2, will release the hormone calcitonin. That's going to cause blood calcium levels to fall and return to a homeostatic level. If blood calcium levels fall too low and they fall below the set point, then the parathyroid glands that are on the back of the thyroid gland will release parathyroid hormone. So the parathyroid hormone then is going to cause blood calcium levels to increase. If blood calcium levels go too high, then we start that process over again. So today we've seen 2 examples of antagonistic hormones. We looked at insulin and glucagon. You should be able to describe how they pair together to maintain homeostasis. And we also looked at how calcitonin and parathyroid hormone pair together to maintain blood calcium homeostasis. So you could use either one of those as a great example when you're describing the value of negative feedback in maintenance of homeostasis or that constant condition. You could also use diabetes as our example of what happens when homeostasis is not maintained. And given the information that we went through today, you should be able to explain what happens when insulin doesn't work properly. So you may want to practice those things prior to your test and claim one of those as your example of how homeostasis is maintained in the human body.